I try to imagine a fellow smarter than myself, and then I try to think, what would he do?
Charge up your axons, ready your receptors, and shift your lobes into upper beta phase. You are listening to Smart Drug Smarts, the podcast dedicated to helping you optimize your brain with the latest breakthroughs in neuroscience, nootropics, and psychopharmacology.
Hello and welcome to Smart Drug Smarts. I'm your host, Jesse Lawler, excited to bring you episode number 83. In this podcast, dedicated to the improvement of your own brain by any and all means at your disposal. 83 is getting to be a dangerously large number. I mean, not really dangerous in any particular sense, but it makes me think ahead to the 100th episode special edition that we have planned and the fact that we've only got 17 weeks before that's right on top of us. I haven't actually looked at a calendar and counted out 17 weeks and figured out when that is, but I'm sure it'll be here before we know it. This week's episode is going to be about Aniracetam, one of the racetam class of compounds. The original compound denoted as an Ampakine, which is now a family unto itself, and my own personal favorite of the racetams. Some episodes before, I did an episode called Jesse's Aniracetam Self-Experiment and sort of recorded myself as I was taking Aniracetam for the very first time. A very non-scientific case study, but nevertheless it was kind of fun, and actually a lot of people have listened to that episode and gotten some questions for a more scientific approach to the same chemical, so you're getting that today. We're going to be speaking with an expert on drugs' effects on the brain, Professor Keith Wesnes, who holds professorships at multiple universities at Northumbria University in Newcastle and Swinburne University in Melbourne, Australia. Swinburne you may remember from past Smart Drug Smarts guests like Con Stow. I think Swinburne has now provided more Smart Drug Smarts interview subject experts than any other single institution. So thank you to Swinburne, and consider this a dare for all you other institutions out there to catch up in providing awesome guests. But we'll be talking with Professor Wesnes in the main interview. If you hang around until the very end of the episode in the Ruthless Listener Retention gimmick, I want to tell you about a study recently completed that attempts to answer the question as to whether humans are the only animal with true self-awareness. And they're finding that the answer is no, and I want to see what you think about the logic that they use to come up with that. So hang around for that in a rather philosophical Ruthless Listener Retention gimmick. But for now, let's start off, as usual, with This Week in Neuroscience.
Smart Drug Smarts, This Week in Neuroscience.
So scientists in Germany at the German Center for Neurodegenerative Diseases, led by Professor Stefan Remy, have recently published findings about a part of the brain that they've identified as essentially behaving something like a speedometer, but also something like an accelerator, keeping track of the brain's physical movement through space and how the hippocampus in particular needs to react to that speed of movement. Because one thing that I hadn't really thought about until I read this paper, but is certainly true, is that the speed that an animal is moving at has a lot to do with the amount of redundancy in the input signals that are going to be coming in from that creature's perception of the world. If you're running down a hallway, you've got information coming at you a lot more quickly than if you're just sitting in one space within that hallway. Humans are maybe one counterexample of an animal where we've actually got these very active inner lives where we could be daydreaming or imagining about things, plotting out plans, and the 3D world around us isn't necessarily providing the only important reason for our brains to be keeping busy. But for animals like the mice in this study, this is really a big deal, and what it turns out is that the actual speed of movement within the motor neurons ratchet up and ratchet down the activity within the hippocampus based on the amount of perceptual information that the brain is apparently going to be needing to process. Says Remy, the electrical activity of the hippocampus undergoes rhythmic fluctuations. The faster we move, the faster certain nerve cells are activated. This increased activation rate sensitizes the brain. It becomes more receptive to the changing sensory inputs that have to be processed when moving. This particular area of the brain is quite small. It's really only a few thousand cells in mice. It's located in an area called the medial septum that is directly connected to the hippocampus. We assume that humans have similar nerve cells, as the brains of mice and humans have very similar structure in these regions. The hippocampus also has major roles in memory, and particularly for spatial memory, being able to keep track of the speed of movement is really important as the brain lays down memories about physical locations. And if you're processing the speed of movement incorrectly, you might be drawing a too large or too small mental map representing the area that you're moving through. Scientists are hopeful that this research could lead to new treatments for diseases like Parkinson's, where a lot of the problems stem from the inability of the brain to correctly control the timing of physical movements. As mentioned, these cells seem not just to be a speedometer for the brain, but also to a certain extent an accelerator. They're found to turn on and control the speed of motion. Says Remy, we found that they also give the start signal for locomotion and that they actively control its speed. Until now, this control function was almost exclusively ascribed to the motor cerebral cortex. Happy to report that we picked up another couple of 5-star reviews in iTunes this week. Gas12345 says, "Keep the science coming, very interesting topics and lots of real-life applicable information." And Murloc18 says, it's well organized and well put with facts and fair overview as opposed to some of the pushing of snake oil and miracle drugs that I so often find. Very factual and straightforward. Thanks to both of you. And moving from iTunes to the website for a second, I also had a really interesting comment that came in off a piece that I posted on the website last week by a reader, Elliot English. There's not enough context here without having read the article to really talk about his response much, but he gave a link in there that was also really interesting, talking about the parallels between intellectual bluster among academics and sexual signaling in peacocks. Which there's a danger, of course, to how far you want to stretch any analogy, but I thought that was a pretty clever one. We'll put that link up on the blog post for this episode also, so you can follow that. Thank you also to all of you who have signed up for the email list recently. Again, we've got two really simple ways to do that. One is simply by going to the website and looking for the fill-in-the-blank email forms, which I think are floating at the lower right and also the very top of the screen if you haven't signed up. And if you're listening on a mobile device, which I know a lot of people do, if you have the ability to text message and you're within the US, you can simply text SDS for Smart Drug Smarts to the phone number 33733. Now, there is a specifically good reason why this week, if you're not on the mailing list, you should do it before next week. You should not hesitate. You should not wait. I've got some news to announce. It's not quite the level of aliens landing on the White House lawn. It is not me announcing that I'm actually a talking dog. I used to be a regular Cocker Spaniel, but I took enough nootropics that now I can host a podcast. It's bordering on that good, but not quite that good. But the people on the mailing list are going to hear about this first. I would like you to be one of them. That's as much encouragement, bordering on obnoxious, as I'm willing to give. But if you want to sign up for the mailing list, you know how to do it. And very exciting news coming out on July 9th. But now let's move on to the main interview.
Smart Drug Smarts.
So we're going to be talking with Professor Keith Wesnes, who's been studying the human brain and its effects from various pharmaceuticals for over 4 decades now. In 1986, he founded Cognitive Drug Research Limited, CDR, which now employs over 50 staff and has offices in the UK, Chicago, and Melbourne. Research findings from the CDR system have been used to support numerous regulatory submissions of new medicines and have been published in leading scientific journals including The Lancet, the New England Journal of Medicine, and the Journal of the American Medical Association. So when it comes to the study of the effects of particular drugs on the brain, it would really be hard to find a more competent expert. We're going to be talking a little about Professor Wesnes' early career and what got him to this point, and specifically about some of his findings on the chemical Aniracetam. We didn't have the greatest reception on the conversation that you're about to hear audio-wise, so I apologize about the quality of some of the audio, but it is well worth listening. So let's jump in with Professor Keith Wesnes.
I started at university in Reading, the UK, in 1970, and I went to do physics, but I found that too theoretical, so I switched to psychology. And for my undergraduate project, my supervisor suggested that I looked at the cognitive effects of nicotine. We didn't know that nicotine had a positive effect on human attention at the time, but there was some evidence he had conducted in work with animals suggesting it was a attentional stimulant. Nicotine was known at the time to affect the brain cholinergic systems. And cholinergic systems, as we know, are the basis for Alzheimer's disease, although that hadn't been discovered at this time. The groups in America and England who discovered Alzheimer's disease did that in 1976. So this was early evidence that the cholinergic system was involved in attention, and the Medical Research Council in the UK supported a PhD project for me to follow up this research using, again, nicotine, but also using a drug called scopolamine, which blocks the cholinergic system, makes you inattentive, causes amnesia, and so on. So during my PhD, I gave healthy young volunteers scopolamine, and the idea was if the cholinergic system was involved in attention, then their concentration, their attention should become poorer, which it did. And then I found that nicotine could reverse those impairments produced by scopolamine. So that was a start of what we call a scopolamine model. Having developed the scopolamine model and published it, the thing I was most interested in was developing sensitive measures to measure the improvements you can see or could potentially see with nicotine and other drugs. So halfway through my PhD, I spent 6 months at the University of Indiana, Bloomington, and they had the early laboratory computers there, which were really exciting. And when I got back to Reading, we had one. And from then on in, I've been automating tests, so presenting information on computer screens and getting people to respond to that information with response buttons, response modules. And that's a very sensitive way of measuring changes in cognitive function. So from the 70s until the early 80s, I developed a computerized test system. And then I set up a research company to provide that system to the pharmaceutical industry and other groups who are interested in cognitive function as a scientific service. Hoffman-LaRoche read my work on scopolamine and nicotine and said, we've got Aniracetam, we'd be interested to study it in the scopolamine model to see whether it would reverse the effects of scopolamine. So we conducted that. We did that trial in Holland with 28 healthy volunteers. So within an hour of injection, scopolamine makes you very drowsy and very inattentive. And then we'd give the compound or the study compounds to see the extent to which they could speed the recovery from this impairment. And what we found was that Aniracetam was very effective in actually reversing the effects of scopolamine. We also studied Piracetam, which was the forerunner of Aniracetam, worked not quite as potent as Aniracetam, but that also had some beneficial effects. But Aniracetam was the most effective of the two and clearly reversed the effects of scopolamine, which would indicate that it could be used as a cognition enhancer in healthy people or as something to treat people who have cognitive dysfunction due to aging or the failing cholinergic system or both.
And the people that you used in that test, were those elderly people or were it young, healthy volunteers that had been given scopolamine?
Yeah, the first few years that I did the scopolamine model, we used healthy young volunteers. We have subsequently run quite a number of trials in the elderly. The elderly are more sensitive to scopolamine. Therefore, a dose of, for example, 0.2 milligrams injected subcutaneously produces the same deficit in an elderly person as over twice the amount of that in a young person. So, and that's probably because the cholinergic system is failing with age and they're more susceptible to the effects. The elderly are obviously a slightly better and more interesting population because that's the population we're aiming drugs to treat age-related memory declines and cognitive declines at. So we have used the elderly as well, but the Aniracetam work we did in young volunteers.
And you mentioned Hoffman-LaRoche. I guess they were the company that first synthesized Aniracetam. Is that correct?
Yes, as I understand it, yeah.
Could you tell us a little about how that drug design work went? Because I know that Piracetam was the original Racetam, and in a sense, the phylogenetic tree of the Racetam family is split off in 15 or 20 different directions. There's a whole lot of different Racetams. Now, how did that process come about?
Just clever scientists who were interested in developing, called them nootropics. That was the idea, to actually have positive effects on cognitive function. Piracetam, as you said, was the first one. Aniracetam was another one. I also studied Tenilcetam, Tenilcetam. They're all of a similar family, and at the time we believed they were having their effects by positively affecting the brain neurochemical acetylcholine and helping its release and sustained release. There are other aspects of brain function which the acetams appear to affect, but that's generally the mechanism.
And that's the case across the entire family?
It appears to be. I'm sure there are some differences, and that is my strong field.
So Aniracetam is the original chemical within a class called the Ampakines. Can you tell us about what that is, what that means?
Ampakines are a class of chemical which affect glutamatergic transmission. The glutamatergic system is very important in brain function, has a lot of roles and involvements, and Ampakines affect a particular part of the receptor, the NMDA receptor, in a positive way, enabling more constant transmission and improving aspects of memory. An American company called Cortex have developed several different of these Ampakines as cognition enhancers, and they do appear to be successful and effective. It is a good route for improving cognitive function.
Aniracetam, I've also read that it's a lipid-soluble chemical. When people are taking it, is it something that should be taken in conjunction with food, in particular with fatty food, so it passes into the body easier, or is that a concern?
It's a really good question, and obviously you want drugs to get in. When we studied them, we gave them orally with a light breakfast, and they were effective. But there might be more optimal ways, and I'm sure the producers of Aniracetam will advise you the best way to take it, or maybe whether to have some fat at the same time or whether to have it on an empty stomach. Those sorts of things are obviously important.
To step back to the scopolamine trial for a moment, it gives rise to the question of these people have sort of intentionally had their cognition hampered by the application of this drug prior to bringing them back up to functional levels with Aniracetam. Have there been similar studies, to your knowledge, about people at their baseline level of cognition that they're operating as they normally would to see what improvements there may be?
So certainly the scopolamine model is an effective way of testing, but in a number of trials we've conducted, we've also given a placebo scopolamine administration and the active ingredients, and we have seen improvements. When we studied the compound I mentioned earlier, Tenilcetam, that did reverse the effects of scopolamine, but also when given it on its own, it did improve aspects of cognitive function. So these compounds don't necessarily require inbuilt or temporary impairment to show their benefits.
That actually sounds like a very well-designed study that you have placebo both for the drug that's acting to impede cognition in one case and then a placebo for the drug to bring cognition back in the other case. It kind of covers all your bases.
It does, yeah. Made writing the paper fun.
I've taken Aniracetam myself on occasion for about the past year and a half, and I've definitely sort of noticed it to have some mood-boosting effects. I've read that in animal models, they have shown anxiolytic effects, anxiety-diminishing effects. Can you talk a little bit about that?
Only a little bit. Certainly, when we measure cognitive function, we also measure self-reported mood states, levels of alertness, levels of calmness, levels of contentment. And certainly, as you say, when we administered the scopolamine, it made them feel slightly more anxious, certainly less alert, and not as content, not as happy as they were. And certainly Aniracetam reversed those effects, so it appeared to have positive effects upon mood as well as cognitive function.
You've done a ton of trials on a ton of different chemicals over the years. What are some of the more interesting findings or surprising findings that you found during that time?
A really good question. One of the first things I studied was ginkgo, and I've always been very impressed with ginkgo, and probably published 10 or 15 different trials that we've run. Also a combination of ginkgo and ginseng together, which was produced by a company called Pharmaton and is still on sale as a product called Ginkasan. 4 separate studies, we showed that improved the ability to retain information in memory. It didn't seem to stimulate them. It didn't seem to improve their attention. It certainly improved their ability to hold and retrieve information in working memory, short-term memory, but also long-term memory. So I've always found that pretty interesting, and for that reason, I take that. Nicotine obviously is very important to me in as much as it started off my research career. And a number of the pharmaceutical companies have developed compounds which imitate nicotine. Nicotine hits a number of receptors in the brain, and they've developed some very specific compounds which are nicotinic agonists, which act in that fashion. We've studied quite a number, and some are under development for Alzheimer's disease, and some have already shown the ability to improve function in patients who are suffering mild cognitive impairments. We did a trial which we published last year with a group in America where they gave nicotine patches to patients with mild cognitive impairments. And over 6 months, we saw improvements in attention and memory in that population. And we're hoping to get a much bigger grant together to use that more intensively. So nicotine itself is helpful, but nicotine obviously has some side effects. It's not very specific. It's what you might call a dirty drug because it hits lots of receptors. And what it does, some very specific compounds which hit alpha-4 beta-2 is one of these specific receptors, which is a mechanistic target. And that does seem to be quite good. And another one is the alpha-7. So they really made these drugs work smartly by just hitting a particular part of a neuronal process, which very specifically doesn't affect the periphery of the body and just has effects in the brain which appear to be positive. So I'm still pretty impressed with those. Antidepressants are quite interesting because they seem to improve cognitive function, and we've studied a number of those. And that is important because depression is associated with very marked cognitive impairment. So it's nice to treat the depression, but that doesn't necessarily get people cognitively fully functional again. And added advantage of some of the newer antidepressants is that they do promote cognitive function as well as treating the depression.
I've heard that some of the antidepressants can improve mood, but cognition can be diminished.
Yes, so the early generations of all of the drugs, so the antipsychotics, the antidepressants, the anti-anxiety agents, were generally sedatives. So you took them, it treated your anxiety, it treated your depression, it calmed you down. They actually impaired cognitive function. So the next second and third generations were targeted to try to avoid that and have different mechanisms of action. One we've studied is reboxetine, which affects a brain transmitter. And we actually showed in patients that compared to an older antidepressant, we saw improvements in the depressed patient's cognitive function. Desvenlafaxine, we've also studied. That's an interesting compound again, and that does seem to have positive effects on cognitive function in depressed patients. So a number are quite effective. And many years ago, we studied meclobamide for Roche. That was a monoamine oxidase inhibitor. And we also found that had pro-cognitive effects in volunteers and also in depressed patients. So there are a number of the newer types of compounds which are more specifically affecting brain chemicals in a positive fashion as opposed to being sedative. And they do seem to treat depression and have a positive effect on cognitive function.
Going back to Aniracetam for a moment, did your studies look overall at any other physical effects, any effects other than the cognitive ones? Are there any known downsides? I'm wondering why it's a prescription drug rather than something that can be purchased over the counter.
That's a good question. All of the trials were very carefully monitored. So they were what we call phase 1 trials. They were done in clinical pharmacology units, which are like mini hospitals. There were very thorough medicals done before and after, vital signs, ECGs and so on. So we didn't see in any of those trials any adverse effects of the compound. So they do seem to be relatively neutral in terms of affecting things which are important to health besides cognitive function. And that's a very important thing, obviously, in any of these trials to ensure, to assess the safety, assess the side effects, report them appropriately, and let people decide whether they were willing to take the risk of the side effect or the discomfort for the positive effect of the compound.
I'm always curious as to what winds up as a prescription drug versus if it's found to be safe is something that can be just purchased over the counter as a supplement. And so that was sort of my questioning there, because I've always heard that the safety profiles on the racetams are actually very, very good. But many of these things in different parts of the world are prescription drugs.
I think it very much depends on the country. But these compounds like Aniracetam, which are manufactured in Europe, they have to be administered under a prescription.
Smart Drug Smarts.
So thank you once again to Professor Wesnes for his time with that interview. He sent over to me prior to that interview a list of some of the compounds that they've tested using the CDR system and the results that they found broken down by the types of cognitive effects that had good, bad, or no particular result, and then further broken down by whether it was attention and information processing, working memory and or executive function, or episodic memory / long-term memory. And they've got, I don't want to say everything under the sun because there's a whole lot of medicines out there, as we all know, but they have done a lot of studies on a lot of different things, 190 clinical trials from 1975 to the present. So don't be surprised if we hear from Professor Wesnes or one of his colleagues again on one of the other compounds in which they'll have expertise. I continue myself to be a giant fan of Aniracetam. I've actually taken it twice this week. It's not a daily use thing for me. I kind of like to hold it in reserve for those times when I'm looking for an extra boost, but actually this was a pretty busy week and I was looking for an extra boost on a couple of occasions. I'll be talking more about my own personal drug stack soon, so we'll save that conversation for later. But thanks again to Dr. Wesnes, and let's move on now to the ruthless listener retention gimmick.
Smart Drug Smarts. Ruthless listener retention gimmick.
So this article caught my eye because it describes itself as a study, and yet it also describes a thought experiment. And this is interesting to me because I'm a giant fan of the concept of a thought experiment, and I think it's kind of neat when people are still coming up with thought experiments now because people have been aggressively thinking about things since the beginning of time, and technically any thought experiment is something that doesn't require scientific apparatuses and gizmos and gadgetry, and Plato could have done, 20-some hundred years ago at this point, but we're only thinking about it now. So it's fun when thought experiments are continuing to break new ground. This one was about consciousness and consciousness in non-human animals, which is sort of a concept I've always been fascinated by, even though it's a frustrating concept to be fascinated by, because the problem with consciousness is that the definition of it is so wavering and so really dependent on the context it's used in and the person that you're talking to. People have definitions of consciousness that vary from what I would consider awareness, like responsiveness to outside stimuli, to a really deep and profound level of self-introspection, and identity concerns seem hyper-specific to humanity at this point. So with that as a long-winded preamble, this is a study conducted at the University of Warwick based on a thought experiment led by Professor Thomas Hills in the Department of Psychology, thinking about what level of self-awareness would be required by animals to essentially plot future actions. The study was published in Current Zoology, but it was inspired by work conducted in the 1950s on maze navigation in rats figuring out how to navigate a maze to find the proverbial cheese, or in this case, the literal cheese. And researchers back then noticed that at points the rats would come to a stop and apparently deliberate what they were going to do, which direction they were going to go. And what the psychology professors conducting this thought experiment did was look at these choice points, as they call them, and offer different descriptive models to explain the process of what might be going on inside the rat's mind. In one model, the naive model as they called it, they assumed that the animals inhibit action during simulation. So they would simulate, if I go left, this happens, if I go right, this happens, but essentially dampen motor control. So much like in a dream, you might think about something happening, but your skeletal muscles aren't acting out those movements. However, they dismissed this model because it would have created false memories that the animal might be unable to tell the difference between real and imagined actions. I imagined going right, I imagined going left, I didn't actually go either, but now I've got these false memories of both things. The second model that they explored, which they wound up settling on, they called the self-actuating model, in which the rats would have tagged real versus imagined experience. I would imagine going right, I would imagine going left, but I would paint these memories with a, hey, this didn't really happen memory, this was an imagined memory. The professors called this tagging the version of the mouse that went down these imaginary paths, the primal self. This was their terminology. And having this primal sense of self, they say, the ability to imagine and yet discount this version of themselves that they thought about at the choice points, requires that they have a corresponding real sense of self to distinguish that primal sense of self from. These are the professor's words, not mine. He says, as such, humans must not be the only animal capable of self-awareness. Indeed, the answer we are led to is that anything, even robots, that can adaptively imagine themselves doing what they have not yet done, must be able to separate the knower from the known. So I read this, I find this was interesting. I disagree with this. I don't know. I think it's a thought experiment. I don't know if I consider it a study. I would love to hear people's opinions on this, and whether you think this is sort of conceptually rigorous, whether you think that real work was done here, or if this is a little bit of intellectual hand-waving. I find myself in the latter camp, but I'm open to argument on that one.
Smart Drug Smarts, the podcast so smart, we have smart in our title, twice.
You heard it. That is the entire episode, number 83, drawing to a conclusion. Thanks for hanging around until the end. If you're still here, I will take that as strong correlative evidence, not completely binding, but you probably liked it. And if you did, if you would care to share Smart Drug Smarts with any friends, relatives, brain-owning Homo sapiens who you happen to come across, we would always very, very much appreciate that. You can do that on social media. You can do that through strategically placed graffiti. You can do that with any number of creative means that I haven't even thought of yet. The webpage for this podcast episode will be up online at smartdrugsmarts.com, as usual, with the links for everything that we talked about here. I would also very, very much encourage everybody to have a great 4th of July weekend if you're here in the USA, but also be safe, be responsible, take care of your brain, save some brain cells for next week. And next week, I will be back at you with a very, very, very special episode of Smart Drug Smarts. I won't divulge what we're going to be talking about just yet, but I will say that I hope you tune in when I'm back, as usual, with an unwavering commitment to helping you fine-tune the performance of your very own brain. Have a great week until then, and stay smart.
You've been listening to the Smart Drug Smarts podcast. Visit us online at www.smartdrugsmarts.com and subscribe to our mailing list to keep your neurons buzzing with the latest in brain optimization. Smart Drug Smarts should be listened to for entertainment purposes only, although some guests on the show are medical doctors, most are not, and the host is just some random guy. Nothing you hear on this podcast or read at smartdrugsmarts.com should be considered medical advice. Consult your doctor and use some damn common sense before doing anything you think might have a lasting impact on your brain.